Okay, everybody. So today I wanted to just have you watch this video just to introduce you what we're going to be learning about in the next week or so. And it's a topic called bioengineering. Sometimes it could be called genetic engineering. So let's just get into it. And like I said, this is just going to be a preview of what we're going to be doing. And if you take a look at these references here, these are for the honors textbook, and this is for the level 2 textbook. This is where the stuff, they get into it a little bit, but your books don't really get into it in a lot of detail, so I'm going to provide some extra detail for you. It's a good question to ask. Why do we need to learn this? Well, before this, you guys were learning about DNA structure, transcription, and translation, and that's the way things are supposed to work. And then after that, you learned about the different tools of molecular biology. You learned about how humans did selective breeding, and that's how we tried to change the DNA of creatures around us to make them have the traits we wanted them to have. And then you all learned about newer technologies that allow us to actually directly change the DNA, like DNA ligase and restriction enzymes, DNA extractions, PCR, which separates DNA fragments, which copies DNA fragments, and then gel electrophoresis, which separates DNA fragments away from one another. This is going to be about the application of those technologies to actually change the DNA of organisms to benefit humans. There are a lot of applications to genetic engineering. You could take an entire course on this, okay? But I'm just going to give you some examples of things that I think you might come across again, either while you're in the high school or maybe in college or just reading about just in your normal everyday life, so you're educated about these things. Okay, the first thing that you're going to learn about as you go through the activities is going to be this stuff called recombinant DNA technology. This is when we combine DNA from two different creatures. And you're going to see, well, why would we ever want to do that, and what benefit would that be? I was going to talk about cloning. Cloning is producing genetically identical organisms from one parent organism. Okay, and this would be creatures that normally reproduce sexually, but we're going to make a clone of a direct copy of one creature. DNA fingerprinting is also called DNA profiling sometimes, and that is going to be taking a DNA sample and comparing it to individuals who you think it comes from to see who owns that DNA. We're also getting into a technology called CRISPR. CRISPR allows us to make direct edits to the DNA of an organism very quickly and very inexpensively. It's a very new technology, and we're going to get into that too. So, just to introduce these to you, recombinant DNA technology is where we recombine DNA. So, recombinant DNA is a DNA molecule that has genes from more than one source. It has its own DNA plus DNA from another creature added to it. An organism that has its own DNA plus DNA from another creature added to it is called a transgenic organism. More so in our society, though, we hear them called GMOs, genetically modified organism. You may have a box of cereal or pasta or a can of something in your house that might say contains no GMOs or something like that. And what that is that company is basically telling you that they don't use genetically modified plants to make that food product, is probably what that means. Genetically modified organisms are not necessarily bad for you to eat. Most brand name foods, if we use cereals as an example, like Cheerios and things like that, they do contain genetically modified oats or wheat in them. And usually they're genetically modified, or they have recombinant DNA in them to allow the plants to survive insects chewing on them, or it may allow them to survive a frost, or may allow them to grow faster. And there's just different ways to genetically modify organisms so that they're healthier, they grow faster, or they have traits that we want them to have. There are some downsides to it, and that's what you're going to take a look at as you do your work this week. Here's just some examples of transgenic organisms. We have taken the gene for a human protein called insulin and put it into bacteria. And now bacteria make human insulin. And why would we want them to do that? Someone who has diabetes, they have to give themselves shots of insulin. Well, when they do that, they're giving themselves human insulin. So we took the DNA that makes human insulin, put it into the genetic code of bacteria, and the bacteria do transcription and translation to that human insulin and just make insulin. Then we can harvest it and purify it. And someone who has diabetes, they give themselves human insulin that's been made by bacteria. I mentioned this one too, that we make plants that can survive frost a little bit easier. Fish that live in the Arctic, they make a protein that acts like a sort of a normal or a natural antifreeze. We put the gene for that into them, and then now these plants, they can make this protein, or it might be more of a lipid sometimes, that allows them to survive temperatures when they dip below 32 degrees. And then glowfish, you can buy these at pet stores. Okay, here's the normal wild type. It's called a zebrafish, and these are glowfish. These are these fish here that have a gene from coral inserted into them. And that's not helping humanity in any way. That's not helping the food supply or producing medicines. That's more of a cosmetic pet sort of thing. But you may have seen that example before. Okay, this would just be an example of producing human proteins and using them as medicine, and then producing plants with different features. Something else you're going to learn about is cloning. Okay, just so we're all on the same page, a clone is a member of a population of genetically identical cells produced from a single cell. So, like I said, this is taking a creature that would normally reproduce sexually and making a copy of one of its cells, one of its diploid cells. And then that becomes its own creature. That's what cloning is. Cloning happens normally in nature. Asexually reproducing creatures are cloning themselves. They're making exact copies of themselves. But we're not talking about the normal, like bacteria cloning themselves asexually. We're talking about a creature that normally reproduces sexually by combining sperm and egg and just taking one of its body cells and making copies of that. And you're going to do some work this week where you'll learn about cloning, the pros, the cons of it. How far along are we with cloning in science, and why would we want to clone things? Okay. Our next topic is probably the newest of the things that we're going to study, and this is called CRISPR. You don't need to know what this stands for, but it's cluster regularly interspaced short palindromic repeats. So, what does that mean? This is very difficult to understand. You're going to watch a video about this where the authors of the video are going to talk about CRISPR, what it is a little bit, and then they're going to really kind of go off the deep end in speculation on how humans will use this in the future. Okay, but let me just explain it to you very simply. Think of CRISPR as you write a paper and you have some errors in spelling or in grammar. You then go in, you cut out the words you don't want, and you paste in the words you do want. That's exactly what CRISPR is. It's a cut and paste type of editing, but it's with DNA. So you cut out a DNA sequence that you do not want, and you are able to paste in those A's, T's, G's, and C's that you do want to have there. Okay, and that's the simplest way to explain it to you. Um, if you think about what I just said to you, all of these things now become true, and some experiments have already been started with this stuff. We could cure genetic diseases. We could cut out the DNA that's causing the cystic fibrosis or that's causing the albinism or whatever it might be and replace it with a good version of that DNA. We could cut out the DNA that causes viral things like HIV or herpes or things like that. Those are viral conditions where the virus puts its DNA into the human host. Well, we could just cut it out, or we could cut it into pieces and that would deactivate it, and then that disease is gone. We could repair those cell cycle genes so that we actually cure the cancers. We don't just remove tumors and try to kill them with radiation and things like that. We can actually go in and repair the DNA of those cells. And Basically, this is what the video is going to get into. The video is a bit sci-fi-ish, but we could change the genetic code in any way that we want. We could cut out genes we don't want and paste in genes we do want. Okay? And this isn't some way off in the future thing. This technology exists. Okay? But the application of it, what we're going to do with it, and how practical it is, that's more of a present and future kind of thing. Just to give you a quick overview of what CRISPR is, basically when certain viruses, there's certain viruses, they just attack bacteria. And what they do is they act like a little needle and they insert their DNA, and then the whole rest of this picture, this is a bacterial cell. Okay? They insert their DNA in there. When bacteria get infected by viruses, a lot of times they just die. But sometimes what they do is they keep a copy of that viral DNA if they've survived the infection. They then incorporate this, and then if they get attacked by the same virus again in the future, what they can do is they can use kind of like it's almost like how a vaccine works. They use this previously acquired DNA to actually fight off this new attack of this same kind of DNA. Okay, and we're basically using that naturally occurring technology to cut out DNA and replace it with other DNA. Okay, so you're going to watch a video about this, and we're going to be talking about this. And finally, you're going to be getting into DNA profiling. It's also called DNA fingerprinting, and that's when we analyze sections of DNA to basically link them to see who owns that DNA. So we are going to take DNA sequences. Say there's blood found at a crime scene or something like that. We'll take the DNA from those cells, probably do some PCR to it to make more copies of it, and we'll do the DNA fingerprinting technology to it, and we'll combine it to who we think left the blood there, to their DNA samples. We use gel electrophoresis, and that actually makes the DNA fingerprint. So these are called DNA fingerprints. It does not have to do with the patterns on your fingers, but just like the patterns on your fingers are unique to you, your DNA is also unique to you. Okay, so what we do is we look at these sections of DNA called introns. We're not totally sure what they do. And as you could see, here is the blood sample found at the crime scene. Here's a blood sample taken from the victim, and here's one from the suspect. And you could see that these little bands of DNA are exactly the same. That's the same. Those two are the same. That's the same. And those two are the same. And that's the same. So what that means is this blood came from this suspect. And that's how we use this in a forensics case. I know some of you are interested in forensic science. We have a forensics course here at the high school. And we will be getting into this just so you get an idea of how that all works. Okay, so that's what we're going to be getting into this week. One thing I do just want to show you before I end this video is that one thing that you're going to need to do is you're going to do a web quest about cloning. And when you do that, you need to enable that Adobe Flash Player. Okay, so remember, click on this little lock up here. And down here where it says Flash, I think it says allow. Okay, but just make sure that it says that. And then when you start clicking on some of these things, you may have to click on where the box is right in the middle of your screen to allow the animation to happen. Okay, and if you're having any problems with that, please let me know. And again, guys, I'm always available. You can ask me questions, email me any issues you're having, and I'll do my best to help you out.